on Earth is known to be high and is thought to be innermost, but the true extent of microbial diversity is largely unknown. New molecular tools are now permitting the diversity of microbes. New molecular tools are now permitting the diversity of microbes to be explored rapidly and their evolutionary relationships and history to be defined. The purpose of this article is to define the scope, highlight major themes in our current understanding of microbial life, and to describe recent progress in expanding knowledge of the evolution and biological significance of these organisms. Types of production. Microbiota present in wheat and semery processing. The microbial diversity of coffee beans in wheat processing is smaller compared with that in dry processing because the fermentation time is shorter, up to 48 hours. Up to 48 height, and there is a more rapid pH decline from 6 to 4.3. This microbiota comprises few bacterial and yeast species. There is no report of filamentous fungi involved in wheat fermentation. The mucilage adhered to beans is the substrate used by bacteria and yeast during fermentation. Higher microbial diversity in washed coffee fruits could be observed using molecular identification techniques that allow the detection of cultivable and uncultivable microorganisms. The molecular identification of microorganisms associated with coffee showed that in semery process, the microbial succession occurred bacteria dominated at the beginning of the process, but after 72 hours, the bacteria and yeast populations reached similar values, average 10 to the power of 6 CFU G-1, and after 192 hours of fermentation, the yeast population predominates. Currently, there is still doubt regarding the apparent bacterial